Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this episode is going out on April the 4th. 2025 seems like it's already flying by. We're almost at Easter already. I'm half expecting Black Friday ads next. It has been a very busy week with interviews for various podcasts. 3 yesterday, and today, the day that the podcast goes out, I have just 7 interviews to do. I hope I don't mix them up, and I also hope I don't lose my voice. It never happened when I was on the radio in Canada, but it did happen once when I was in a band. I didn't actually know that I'd lost my voice until it came to the first of my lead vocals, when the first line sounded a bit like a sea lion barking, although maybe that was an improvement. There were lots of listeners again last week to the podcast and some very nice comments. So thank you, greatly appreciated. And of course, please do get in touch with guest suggestions, which you can send to jim@deeptechdigest.com. Today we have 2 interviews again. One with Sean Rothwell, CEO of EVŌQ Nano, about the development of nanoparticles that can destroy bacteria. But first, we are talking about Satellos Bioscience's work tackling Duchenne muscular dystrophy. The company is developing small molecules to improve the treatment of muscle diseases and disorders, and it recently announced initial Phase 1 data at the 2025 Muscular Dystrophy Association Clinical and Scientific Conference in Dallas, Texas. To tell us about that presentation and what the future may hold for patients with Duchenne muscular dystrophy, we chatted with Satellos Chief Scientific Officer Phil Lambert. Easy first question then is if you could give me a bit of background on the company.
Yeah, so we're a small biotech company, around about 25 people, based out of Toronto, Canada. It's a very experienced team. I joined the company coming up for 3 years ago. I've been in the biotech pharma space for a long time. And what certainly attracted me to the company was that back in the early 2020s, the company really brought the science to bear to the point of discovering a target that we could really go after with a drug to drive regeneration of muscle. And that, along with a group of people that have always been, but continue to be extremely focused on delivering for this patient population. I felt like there was the essential things you need for the potential of success. This is a hard business, as you well know, coming up with new therapies, particularly for complex diseases. And I felt like, the wealth of science that is behind Satellos, because they've been working on this approach and the company's been around for some time. But that, along with a target that somebody like me, a drug discovery and development guy, could go after, and the right group of people to really bring this forward, I thought we were in great shape. We have been fortunate over those 3 years that I've been with the company to be able to raise a couple of rounds. We've done 2 rounds of financing in what I would say most people would say some tough times to raise money. I think that's reflected on the team we've put together to move this forward, as well as the fact that more recently, the science behind what we're looking to do has really come more to the forefront. More people outside of just ourselves saying, you know what, I think this really is key in muscular dystrophies. And if you could allow these patients to regenerate muscle in a healthy way, shall I say, or at a level that you see in healthy individuals, that would really make a difference to them. So we're excited where we are as of today. A lot of hard work, a lot of time, as always, with tracking down complex science has certainly gone into it. But we're excited, where we are today as a team.
I guess what you're doing has quite a lot of wide applications. What areas of disease are you specifically focusing on?
Yeah, you're absolutely right. Our focus for sure is on muscle and regeneration, currently at least specifically of muscle. So that does give us a wide number of dystrophies potentially that we may be able to help. We don't believe that all dystrophies have a deficit in muscle regeneration, but we're certainly aware that there's more than one that does. We are currently laser focused on Duchenne muscular dystrophy. That is a rare genetic disease, the most common, muscular dystrophy. But we have data that certainly indicates that the drug may be useful across a range of muscle diseases. And that's currently where we are laser focused.
Could you maybe go into a bit more detail about Duchenne muscular dystrophy?
Absolutely. So Duchenne it's a rare genetic disease caused by genetic mutations which result in the loss or reduction in function of a protein called dystrophin. And it's an X-linked genetic disease. So the vast majority, almost all patients are male. And the function of dystrophin was identified a long time ago. Folks identified that dystrophin was a key structural protein in muscle. So without dystrophin, muscle is less stable, muscle breaks down more easily, and so you get injury to muscle. Michael Rudnicki, who's our scientific founder out of the university, he's a professor at the University of Ottawa, discovered the key piece, which is really the underpinning behind Satellos, which was that dystrophin didn't only have a role in the muscle fiber, but it had an important role in the muscle stem cell. And this really approached a question that we really hadn't been able to answer based on dystrophin being in the muscle fiber, which is, even though you had a lot more damage in the muscle because of this lack of a structural protein, why ultimately was muscle completely lost? Because in Duchenne, what happens to these boys, most Duchenne boys are diagnosed around the age of 4 or 5. And then over the course of the disease, which typically goes through to sort of the teenage years, the early 20s, they physically lose muscle. Muscle is replaced with fat and fibrosis, and muscle disappears. And it wasn't completely clear why that would occur if the only problem was this loss of a structural protein in the muscle fiber. But what Michael discovered was that actually dystrophin had this other role within the muscle stem cell. And there, rather than being structural, it was actually a signaling role. And what was important about that was if you lost dystrophin, those stem cells were unable to divide in the way they need to produce the necessary progenitors to regenerate muscle. So in a healthy individual, every day, just walking down the street, certainly exercising, we damage muscle. And without thinking about it, that muscle is repaired. And it's repaired by activation of these stem cells. These cells sit on the muscle fiber, don't really do anything unless there's a damage signal. And then they go into action. And they divide, we refer to it as asymmetrically, which means they divide to form two different types of cell. One stem cell to repopulate the pore, a second cell, which is a progenitor to regenerate muscle. And without dystrophin in the Duchenne boys, the efficiency of them being able to do that is very much decreased. So as a damage signal comes in, those cells are trying to go through asymmetric division, but they really can't. And therefore, regeneration of muscle doesn't keep up with the damage. And in a situation where regeneration doesn't keep up with damage, you physically lose muscle. And of course, that has, very, very profound effects on function. So what we were able to do, what we believe we have done, is not replace dystrophin into that stem cell, because practically that would be very challenging to do, but actually come up with a small molecule treatment that actually replaces that signal. It goes in and replaces the signal that's lost with the loss of dystrophin. And now, in response to damage, allows those stem cells to go through that asymmetric division, produce progenitors, and regenerate muscle. So we're not really, we're very different. We're not working on the side of slowing the damage. We're more working on the side of actually allowing the system to respond appropriately to that damage, push back against that, regenerate muscle. And we believe not only prevent the disease from progressing, but in situations where a patient's already lost muscle, potentially bring muscle back, recover function, because that muscle will be regenerated and recovered in that situation.
Before the work that you were doing, what were the standard treatments?
Yeah, there's a large group of people have been working on trying to come up with treatments for Duchenne, and specifically in the case of Duchenne, by replacing dystrophin into the muscle fiber. So gene therapy and what we refer to as exon skippers, trying to recover that function of dystrophin in the muscle fiber. And there has been some progress in that space. Whereas I say we're different is Michael was the first to show that there was another component to the disease, which involved dystrophin in the stem cell. And so we're the one company that's come into the space and said, we're going to push from the other direction as well. And we've never ruled out the possibility that a combination therapy where you need to approach both of these issues, because there's no doubt that dystrophin does have a role in the muscle fiber. And if you were able to effectively replace enough dystrophin there, you could make a difference for the patients. But there's another aspect to it that we feel is at least as important, which is on the regeneration side. Yeah, mainly it's been up until now focused on that damage that comes from dystrophin being lost, or the function of dystrophin being lost from the muscle fiber.
The lead candidate that you're using to treat Duchenne, what the mode of action, what does it do in terms of how it's helping people? Is it helping slow down the loss, or is it replacing muscle?
Yeah, that's a real good question. We believe that it's going to ultimately allow muscle to come back and allow function to come back. What we're focused on preclinically, Jim, is to always say, this is a complex process. It involves a lot of things happening. We're driving the very front end of that in terms of creating progenitors. But what we want to see and what patients need is a return of function. And when we look preclinically at what this molecule does, it is a small molecule. So it's a novel chemical entity that we have designed and developed. It's a Satellos compound, and it acts on a kinase called AAK1, or adaptor-associated kinase 1, which is part of the NAC family of kinases, or NUM-associated kinases. And as I mentioned, ACK1 is within the stem cell, and by a lot of work done through the Satellos labs, screening to see which targets, if we influence them, which targets could allow us to recover this signal in stem cells, to allow the cells to go through asymmetric division. We discovered ACK1. And so if you inhibit ACK1 and then the stem cell receives a damage signal, it's still very important that these two things have to come together. You still need a damage signal coming in to ask the cell to divide. And then we inhibit ACK1 with 3247. We see muscle recovered, and actually, more importantly, maybe ultimately function returning. So we see the force that can be driven by muscle improved. And so, as I say, that came from a lot of work was done within Satellos, looking at all sorts of targets before we discovered one that not only we believe and we're seeing is very safe to go after, but is a very effective way to replace that signal in the absence of dystrophin in the case of the Duchenne boys and drive muscle regeneration. And we identified that as a lead molecule. We have a lot of chemistry behind this, a lot of very interesting chemistry and interesting ways that we worked out how to identify a very selective and very potent compound that acts at AAK1. So we have a number of different molecules, but we identified 3247 for a number of reasons, safety and efficacy reasons, as our lead clinical candidate. And that's the one we're moving forward.
Looking at the effect on muscle long term, does it make any difference at what stage the intervention is? I mean, if you get somebody that has a very minor amount of muscle loss and somebody who has had a lot, can you get them both back to the same stage?
It's a real interesting question. And to be honest, Jim, I think one that we won't know the full answer to until we do some more work in patients, only because we have some reasonable animal models, but one of the things that the animal models don't give us is really that progression of the disease and those various stages of the disease that are critical in people. The way we think about it and what our data support is this idea that as long as you still have functional muscle in an area, because as you say, it's not only different stages across patients, if you like, but it's different stages of muscles even within a patient as they go through the disease. As far as we know, as long as you still have functional muscle, as long as a muscle hasn't been completely lost and replaced with fat and fibrosis, it's likely that you still have functional stem cells. And as long as those stem cells are there, we believe we can drive this process of improving the efficiency of muscle regeneration and driving a positive effect on that muscle and on function. So under those circumstances, it means we feel good about having the potential for an effect across the full range of patients that have Duchenne. But as we were talking about it, it's a very heterogeneous disease going all the way from the younger patients, the children, where they still have very significant function, all the way through to the later stage patients where muscle function has been lost and they have certainly some muscles where it has been replaced very significantly by fat and fibrosis. And that you could imagine is going to be a harder piece to push back against. But one of the things that we feel very positive about with our compound, not only is it a small molecule, will need to have and does have, as far as we know at this stage, a very good safety profile, which is good for patients. It's also very straightforward to take. It's an orally available pill, so it's very straightforward to take. But we believe, some of these other therapies like gene therapy, you have to give at a certain age to have the best chance of effect. And we believe we certainly won't be too pigeonholed here in terms of the breadth of patients that we believe we can help. And we believe the process is somewhat self-regulating, Jim. So, if you push back enough against the damage and there's no longer damage, the process will just sort of settle right there. As long as there's damage and you can make sure that the regeneration is effective enough, the process should roll. So this would be a drug that you would take for a patient would take for the rest of their life, because it would be important to maintain the effectiveness of muscle regeneration to push back against any damage that was occurring to the muscle.
You recently came out with some phase 1 data and you presented that at a conference. Could you tell us a bit about that?
Yeah, so we've completed a pretty standard phase 1 SAD/MAD, so single ascending dose and multiple ascending dose, looking specifically at safety and PK. Safety, obviously the key thing that you have to do, and the results for that were that we really have no safety issues with this compound up to doses that are well above where we believe will be for efficacy based on all the data we have. As far as PK was concerned, there was an added aspect to why PK was important to us. First was just to make sure that the compound behaved well, and it does. We saw dose-related PK. We saw no accumulation of the compound at doses at the sort of efficacious level, which is, important. But the second piece around PK that was also key was we understand the PK we need of the molecule, sort of the length of time and the level it needs to be at on the target to get efficacy. And we really wanted to see if it translated into people, that we could obtain that profile in people at the sort of dose level that we were expecting. And the answer to that was we could. So we believe for efficacy we'll be at a very reasonable dose level. We're all set with our CMC to have those tablets available, those pills available to move forward into phase 2. And we achieved the profile that we believe should give us efficacy. A second piece to that phase 1, which is ongoing right now, is our first step into patients. We have not yet reported any of that data because we don't have it quite yet. The study is ongoing as we speak. And the reason we stepped into a small group of patients initially, basically for the same reasons in healthy volunteers. We wanted to check safety, but we also wanted to make sure that there was no difference in that PK profile between healthy volunteers and patients. We do have some, if you would call it, efficacy readouts, functional readouts coming from that study, but it's only 28 days of administration, and we're not expecting to see too much happening in those patients after only 28 days. It's a very short period of time in a disease like this to be treating, and based on what needs to happen for muscle regeneration. But we do have some readouts coming from those patients that allow us to take an initial look. Our clinical team is working very hard to put together the pieces around our phase 2 program, which would be the next step for us. We're hoping in Q2 we will have the data from those initial patients, and that we will also be able to release our plans going forward for the phase 2 studies.
And what's myoregenics?
That's a very important assay, and everything I've just been saying really. So if we go all the way back to the earlier days of Satellos, when we were looking for a key target, a target that we could inhibit, in this case, that would actually return the signaling. We were able to do screening in myoregenics, which is an assay that was developed in Dr. Rudnicki's lab at University of Ottawa, which basically allows you to take muscle fibers in a dish and still maintain the ability to look at stem cells in action. That's really the bottom line. So it's a way to be able to see and look in a dish scenario, in a ex vivo, I suppose it really is. You take the muscle fibers from the animal, or potentially even from a person, and you can actually run the assay and look at how stem cells are behaving. And that allowed us, using an siRNA screen, to inhibit a large number of different proteins that we thought may be involved in this process. It was actually how we identified AAK1, because we could look at the impact of genetic manipulation or compounds in this assay and their effects directly on those stem cells and on asymmetric division. So it's been a key assay. It's an assay that Dr. Rudnicki set up and developed in his lab, and has certainly been important to us in terms of the screening process for us to come up with the target that we're now going after.
You mentioned where you're at kind of with ongoing trials and where you hope to be. What are your hopes, I guess, ultimately for what this means for people suffering with Duchenne?
Yeah, we really believe that it could be a transformative medicine, Jim, in this space. And it comes down to sort of what you mentioned about where patients sit in the course of the disease. Up until now, the treatments that people have been looking to come up with really would slow progression of the disease. And obviously that would be significant for patients, but really very significant for those patients that you can treat as early as possible. There are a lot of DMD patients sitting out there who are further along in the disease process. And what we think we can do here is to not only maintain, but improve the function that they have. And as far as it being life-changing for these patients, to allow them to continue to do certain things by maintaining or improving function for them in their muscles, would be very, very significant. Where that lands us ultimately in terms of being able to help patients regain function, as I say, we really won't know until we play this out in the patient population. But, my goal as the CSO coming on board at Satellos was to put us in a position where we identify a safe compound that would act on the necessary target to go into people and test this mechanism in this theory, Jim, right? We needed to be in a position where, because we can do as much work as we want preclinically, but ultimately we need to know if it translates into people. And very often that step can be challenging. So if we have a safe molecule that will do what we need it to do, at least from what we understand from our preclinical data, we can take that and put that in patients with a very little degree of risk for those patients, but with the ability to test that mechanism. And I think that's right where we sit today. That's where we are as of today, based on the data we got from our phase 1. And now, we go through the process of designing how we're going to approach that in a phase 2 setting. We're already, in terms of availability of drug substance and drug product, we have the compound in hand to be able to do that. And now we're just very, very excited about putting that into patients and seeing how much we can really drive back muscle for them and function for them. But it has the potential to be a very transformative medicine, initially for us here in Duchenne, and then hopefully even potentially more widely in some of the other muscular dystrophies as well that involve damage to muscle. We may also be able to help.
Lots of work still to do then on lots of different.
Yeah. No, it's a busy time. We have a fantastic clinical team, a very experienced clinical team they're very much in the midst of putting all these pieces together, and we can get the compound to patients. What I've always liked, I've been involved in a lot of small companies. I'm a biotech guy in terms of starting up small companies, and it's amazing what you can do with the small group of the right people. And we've always been focused on doing everything, appropriately, correctly that we need to do, but moving it as efficiently and effectively as we can, because we know You've heard this from other folks, but it is important. Patients are waiting there for something to help them out, for something to improve the situation for them when they have Duchenne. And we're trying to get there as quickly as possible so we can, hopefully help them out. Maybe the last comment to make, our preclinical data really supports the idea that the longer we treat, as we reset this whole system and get the system back to where it needs to be, the better the effect will be as the regenerative capacity starts to match up more appropriately to the damage. So we don't think our trials will have to be too long to show something, but we certainly also think that as we continue to treat patients, it potentially should help them more and more over time. And so we're going to try and get as much information as we can, as quickly as we can, to support that and ultimately get the drug into as many patients as we can.
I always find these conversations really uplifting when there's so much optimism about treating a disease. Of course, just like the company, you want it all to happen as quickly as possible, but definitely these are all steps in the right direction. Right, now we are going to talk about nanoparticles. Well, I'm the one doing the listening, doing all the talking is Sean Rothwell, CEO of EVŌQ Nano, whose nanoparticles are tackling a whole host of things. The company's nanoparticle is already in development for an inhaled therapeutic for cystic fibrosis patients, as well as for antimicrobial medical devices, and for the treatment of the surface of textiles. It's also being earmarked for applications in energy and agriculture. And Sean has all the details. First things first, then I wonder if you could give me a bit of background on the company.
Yeah, EVŌQ Nano you're going to hear a little bit of boldness in this conversation because there's all kinds of different nanomaterials out there in the world. There's been a lot of deep research that started kind of back in the early 70s around nanotechnology, recognized that these small atom type size materials could have an impact on a lot of different elements in the world. So, there's been a lot of talk around silver and gold nanoparticles and the such. We've really broken the code of how to create safe, stable, scalable nanomaterial. That's quite a unique statement when you think about nano, because if you look at the broad research that's been done in biotech or in any other verticals such as energy or agricultural or electronics, the common theme of where nanomaterial has really not reached its full potential is the ability to create a nanoparticle that is stable. Stable meaning it doesn't break down and fall apart easily. Secondly, something that is not just spherical, like a kind of round, but actually a perfect sphere. That provides a really smooth surface area for there to be a lot of activity with the nanoparticles. And then the ability to actually manufacture those particles in a specific size range for a specific application. And then fourthly, being able to manufacture those particles at scale. And EVŌQ Nano has been able to solve all those problems. William Niedermeyer, our founder of the technology, discovered a high energy process and very customized laser process of ablating any metal on the periodic table to create these nanoparticles that are 10 nanometers and smaller, that are perfectly spherical. They don't fall apart, meaning that they're stable enough that they can put through high heat, high pressure scenarios and not degrade or fall apart. And when you're using that in a therapeutic, that's critical, because when a particle breaks down into something else and becomes a new element, there's all kinds of toxicity concerns and issues around that as well. And then being able to produce it at a specific size range that actually is efficacious with a specific application is really critical as well. So EVŌQ Nano has been able to accomplish consistent manufacturing of perfect round spheres in any metal at a very high rate of production. For example, one of our verticals is a textile vertical where we're treating all kinds of large textiles all over the world, that are going into clothing, garments, towels, sheets. And so the ability to create this stable nanomaterial has really been a big move into the biotech space. So when we're talking now about what we're doing in biotech, we've got two things going on right now. And one is a therapeutic that we've looked at a cystic fibrosis indication of being able to take these particles and use them as an antimicrobial or anti-infectant to kill bacteria infections in the lungs. And I'll tell you a little bit more about that, Jim. But secondly, we're using this material to be able to synthesize it right into the medical polymers that are being used for catheters and other medical devices. And we've successfully achieved that as well now. So we've had a lot of success with this. Our asset in the bio vertical is called EVŌQ 218. And this material is being used in those two fashions currently, and we're having really good success. We're in commercial conversations. Stay tuned because it will become public here in the next few weeks with some of the commercial arrangements that we have in the medical device space with some of the largest manufacturers and OEMs in the space, to be able to do what's never been able to be accomplished before, is to fully synthesize an antimicrobial nanoparticle through the full thickness of plastic and create a lifetime effect to where the bacteria cannot grow and biofilm cannot form on the surface of a catheter or other devices that we're applying this to.
Is this something that other companies have worked on, or is this completely new?
No, this is something companies have worked on for years and years and years. For example, in the catheter space, you can find multiple antimicrobial agents that have been used, and it's typically silver with a lot of different chemical compounds. And the problem, let me give you that example. That's a great question, Jim, because if we're talking about specifics in the medical device space, when silver colloidal silvers or silver chlorides are used, those particles are not stable enough to actually be used as a standalone property. So how typical silver actually kills bacteria is that silver breaks down and emits ions, and those ions are what destroy the surface or kill the bacteria. And those ions are the same reason that the silver is actually not super effective, because one, those ions are cytotoxic. They create cytotoxicity. And secondly, they agglomerate inside of the human body in different cells and tissues and organs, which is not safe. With our particles, first of all, those coatings that have been being used in the past that I'm referring to, Jim, they're actually coatings that just go on the surface of a catheter or something of that nature. And they're not stable. They don't have a long shelf life. And ultimately, the cytotoxicity creates an issue as well with many of the patients that are using them. Our particles, and the difference with our particles are actually not emitting ions. So when I talked about stability in the beginning, this is a really, really big deal about this breakthrough. We do not have any oxidative layers on the surface of our nanomaterial at all, meaning they're not going to emit ions and they don't fall apart and break apart. That brings us to another scenario where the belief that the only reason silver is antimicrobial is because of this ion emission. And that's commonly accepted, understood, and known in the medical community specifically. And our particles don't emit ions. And so then the challenge is, well, how are you killing the bacteria? Because when we did the studies with our therapeutic, our inhaled therapeutic, the Cystic Fibrosis Foundation gave us 2 grants. One was to see if we could kill these drug-resistant bacteria, and 2 was to see if we were safe. So we killed 64 out of 64 isolates from the 9 most drug-resistant bacteria, 100% kill. And our method of action is not emitting ions. We're actually disrupting the metabolic process within the bacteria cell, because sulfur is attracted to the silver, and the particles are small enough to penetrate the cell wall without damaging it. And we're halting the metabolic process within the bacteria cell without lysing the cell wall, which then leads to another big discovery. We've gone through 3 serial assay passage tests, one for 28 days, one for 35 days, and just finished the 3rd one going head-to-head with the 2 most powerful antibiotics on the market. And after all those studies, we show 0 resistance from the bacteria, where even the leading antibiotics that I'll leave nameless until we publish this, they are showing resistance. The best one showed resistance at about 9 passages in, and ours went all the way through. All those assay passage tests was showing 0 resistance from the bacteria.
You mentioned it in the capacity of with medical devices. Does it have the ability to, like, antimicrobial resistance is not just an issue in medical devices, it's an issue with disease and all kinds of things. How does it work in that capacity?
I'll finish that analogy too that I gave you with the coatings, the silver coatings that have been historically used with catheters and other devices. Our material has successfully been incorporated right into the manufacturing of the plastics. And in addition to, we also have worked with some hydrophilic and antithrombosis coating groups where we can do a coating, but we've integrated right in the manufacturing the process of the catheters and the medical plastics, where typical silver could never withstand that heat or process of manufacturing. And so with that application, we know and we're thankful that we can make the statement that we're never going to contribute to drug resistance or AMR getting any worse when we're doing this in the medical device space. When it relates to infections, and like for our pulmonary therapeutic with EVŌQ 218, we know that we're killing drug-resistant bacteria, which is the death sentence for a cystic fibrosis patient if they get infected with a drug-resistant bacteria and that heavy mucus in their lungs. We know that we're actively penetrating the biofilm within that sputum in the lungs and getting into the bacteria to kill the bacteria. And the big extra bonus to this is we know that the bacteria are not forming resistance to our particles because the method of action is so unique, it's so novel. We're not lysing the cell wall and opening up the communication channels for those bacteria cells to continue to mutate and form resistance in their biofilm against our particles.
So what happens to the bacteria once they're unable to function?
The bacteria simply dies. It's fascinating to watch time-lapse videos of our particles. You can see our particles actually penetrating the cell wall of a bacteria. You can see this attraction, this affinity that the sulfur has within the cell to our particles. And that sulfur really acts as a transport for energy production inside of the bacteria. The other thing that we notice is our particles is just not a one and done scenario when it enters into a bacteria cell. They will continue to follow the fluid paths in the body or in that area. You can see our particles will go from bacteria cell to bacteria cell and actually halt the metabolic process within that cell, and therefore the cell just dies.
You mentioned that therapeutic effect. How is it delivered in that sense?
When we're looking at dealing with infections in the respiratory system, our indication and combination will be our particles with a nebulizer. A mesh nebulizer will be used to inhale the material. When we get into deeper clinical studies and trials, we will certainly be adding some indication for intravenous use of this as well. But we will begin with the inhaled therapeutic, utilizing a mesh nebulizer to get that material into the lungs, which we've already done some preliminary animal testing for safety and toxicity, and the results look really, really good. We're seeing those particles, rather than agglomerating like a typical nanomaterial would because it falls apart, we're seeing our nanoparticles follow the natural fluid paths of the body and are excreted through the natural fluid flows of the body.
In terms of other applications, is it something that could potentially be used? And I guess another thing where infections is really critical is in treating wounds.
Absolutely. Yep. We've actually got statements of work with companies. We're working on different wound care applications and personal protective equipment, the PPE equipment that's being used in the medical facilities. That comes into play with our other division called EVŌQ. EVŌQ is actually applying this to textiles and surfaces. We've got 3 EPA registrations in doing that. So there's applications with that as well. We also are actually doing work in the energy field because our particles do not break down again, and they're these perfect spheres. We're doing work with energy and battery applications where studies have been done in the past It's fascinating, Jim, because the studies that have been done in the past in creating this unique nanomaterials or gold nanotubes or there's a variety of things being used. At the end of the studies, it works great in the lab, but just to create the stability for those particles to go through testing criteria in the lab is so expensive, so arduous, there's no way for any commercial application. Where our particles don't break down and fall apart, we've had tremendous results. And I promise you will hear about this in the media over the coming years. We've had tremendous results in actually enhancing batteries and enhancing different energy functions for our use, utilizing some different types of nanomaterial. So again, we can make this nanomaterial out of any metal on the periodic table, and the application and use cases are so broad. And that is my message and announcement, is we've been able to create safe, stable nanomaterial at scale, very cost effectively, and we can produce this in any specific size range that we're looking for a specific application.
What would the different sizes be relevant for?
The different sizes are absolutely relevant for the use case with regards to what you're doing. For example, when I'm talking about using this as an inhaled therapeutic, the size range of materials, there is a Goldilocks range where you get very, very high efficacy with these really stubborn bacteria. And where you're talking about an application in textiles, for example, you're talking about a larger size range that has very, very strong adhesion to the material that it's being applied to and still has a very broad efficacy rate with all of the bacteria that it would come in contact with to create an anti-odor or antimicrobial clothing device. So size ranging is critical from the standpoint of which application it's used. So there's a lot of science here, because it's not just saying, hey, we just figured out how to create these stable particles. By the way, we've got 58 issued patents to date. We have 50 additional filings in right now. Not only can we create this really stable, scalable nanomaterial, but we can specifically create it in size ranges for specific applications with any metal on the periodic table. So the use cases for this are so wildly exciting. The mission and purpose for me in this is we need to make sure this nanomaterial gets out into the world to do all the good that it can do. Because the research that has been done, I've named them in the beginning of the conversation and brought up several more. Those 4 failure points of your nanomaterial that's been being researched now for 50 plus years, we've solved the gaps and the problems from those issues of stability, size distribution, no oxidative layers so the particles are not breaking down, and the ability to manufacture at scale to where it's a commercially viable opportunity. We've solved those problems, and the future just looks incredibly bright for us.
Just going back to what you were mentioning about an inhaled example, does that then stay in the body forever, or does it pass out of the body?
No, your typical nanomaterial, the reason it's never been able to even be considered for that is because it does stay in the body. Our material will not. I'll be really concise about this part of our conversation. So the Cystic Fibrosis Foundation, they gave us the grant. The studies took place up at Seattle Children's Institute to see if we could kill these really drug-resistant bacteria. When the studies started, the scientists, I remember them saying, if you were able to even kill 1 or 2 of these drug-resistant bacteria, we would be so excited about this. So at the end of those studies, we had studied against 9 of the most dangerous drug-resistant bacteria that exist, named by the World Health Organization and the CDC, with 64 different isolates, and we had 100% kill. They came back to us and said, this is too good to be true. You've got to be severely toxic. There's nothing that kills these bacteria 100% without there being some severe toxicity. So they gave another grant for us to do studies with animals. So a third-party lab put an inhalation chamber together to have rodents inhale our material at 60 times the dosage we would ever use in a therapeutic for humans. They had them inhale this material for 7 straight days. And at the end of the 7 days, there were no deleterious effects to the rodents. Then they do the post-mortem and look at where all this material went in the rodents' bodies. And it was very evident that our material just simply follows the natural fluid paths of the body, and the body excretes our material. And again, these are perfect spheres. There's not any hard edges on them. They don't fall apart. They don't break apart. So there's no agglomeration in the lungs, the cells, the tissues of the body. We actually saw that in our animal studies, that material is leaving the body with the natural fluid paths that flow through our system, our renal systems, our respiratory system.
And I guess one of the biggest things with antibiotics has always been that it's been relatively inexpensive. How have you addressed both scale and cost?
Cost is not a problem for us to manufacture. Think about what I described on the textile side. If you went into my warehouse downstairs, you would actually see container loads of this material going to over 200 factories in the world treating textiles now. We can create that kind of scale. And the material, there's a different size distribution, but the material in the manufacturing process is the same. They're perfect spheres, again, going through the stability factors I'm talking about. The amount of material that we actually have to use to create this antimicrobial effect, it's not cost prohibitive at all. Matter of fact, when we're talking about medical devices or a therapeutic, that's been some of the original questions that you just asked me come from the OEMs and the pharmaceutical community, is like, how cost effective is this? It's exponentially more cost effective than most of the drugs that we have on the market from a manufacturing standpoint.
Where are you at with the development in the various areas that it's going to be used in?
In our therapeutic right now, in our health therapeutic, we're in our IND enabling studies. We are actually through the pre-IND stage with the FDA. Things went really well with that. And so we're doing our IND enabling studies to move into human clinical trials. So we anticipate we're going to run a phase 1B, 2A clinical studies, and we anticipate those starting probably close to Q2 in 2026. So we're about a year out from that. With our medical devices, we actually will be announcing some commercial deal structures here in the coming weeks and months with being able to get medical devices embedded with our material and get on the market. And I anticipate that we'll probably see some devices on the market in 2026 in the catheter division, and there may be a few other that sneak in there with it with some of the partners we'll be announcing. With our textile and surface treatment division named Fuse, it's starting to grow incredibly well. Our EPA registrations were granted in 2024, and that division is it's really, really growing fast. Like I said, we're in 200 factories around the world in 17 different countries, and big brands, like really big brands. And I'm not sure how much liberty I have to name the brands, but we've got very big brands that have adopted this technology because we're getting rid of these forever chemicals by abusing our material. We're getting wash tests over 200 washes where our material is still present, creating an antimicrobial effect. So we actually did get a lifetime of the product claim from the EPA when we apply this to textiles. I'll shift gears and talk a little bit about energy. We're actually in the process now of working out some different new prototypes in the battery space with a different material that we're using in battery to enhance the lifespan and the power density of batteries. We're just seeing tremendous results. There's so much research that's been done in the past, Jim, about how you can use nanomaterials, for example, with batteries. And those failures that I mentioned early on and multiple times in this conversation are consistent of why outside of a laboratory, there's no commercial viability for nanomaterial with batteries on a really large scale, where we're showing that we have commercial viability on a very large scale in lead acid batteries, in lithium ion, and in solid state graphene batteries. So those are where our studies are taking place, and we're actually in prototyping and testing mode with that. So we're hoping that we can have a breakthrough with some of the licensing arrangements that we're working on in that division. Those are some highlights of what's going on with EVŌQ Nano and some of the assets that we're actually building and are really, really close. The timing of this conversation is when I have this conversation with you or others, it gets more and more exciting because we're getting so much closer to that goal line where we're commercializing some of this rather than spending all the years and work and time in the labs and in the study rooms doing R&D and perfection through the process.
Is there anything else that we didn't talk about that you wanted to mention?
I mean, I'm so excited about our asset EVŌQ 218, which is being used in our biomedical and our therapeutic pursuit. When Alexander Fleming gave his acceptance speech of the Nobel Prize back in 1940 for creating penicillin in 1928, he warned us. He warned us that this was a temporary solution because bacteria would form resistance against those types of antibiotics. He warned us. And that time is here. And the serendipity that I can sit here and say to you is we've discovered the ability to create nanomaterial that can hopefully come in and create this new realm of medical applications where bacteria can't form resistance against it. And we have something on the market worldwide that is safe and efficacious for people, where these drug-resistant bacteria don't become what the World Health Organization is calling the existential threat to humanity. That's how they look at these drug-resistant bacteria, and the list is growing. So what we're doing, it gives me goosebumps. It gives me a high sense of feeling of responsibility. But I'm so excited about that asset EVŌQ 218 because it comes right down to saving lives, reducing infections in the healthcare environment, and actually being able to have a therapeutic that will kill drug-resistant bacteria, and new bacteria can't form resistance to it.
Really interesting information there from EVŌQ Nano. Seems like there are so many different applications and that the results are really exciting. Definitely a case of watch this space as the company moves this forward. Right, that's it for this week's wide-ranging interviews. So until next week, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next week for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.